Namaskar. Welcome back to the lecture series on sericulture. In this session, we are going to see the regulation of silk encoding genes. Before we go into this one, let's see how silk is formed, and later we can see the genes that control the silk formation. Silk is a continuous strand of two filaments cemented together forming the cocoon of silkworm, the Bombyx mori. Silk filament is a double strand of fibroin which is held together by a gummy substance called the silk sericin or silk gum. Silk fibroin is the protein that forms the filament of the silkworm and gives its unique physical and chemical properties. Silk adapts various secondary structures including alpha helix, beta sheet and cross beta sheet. Fibroin is a glycoprotein composed of two equimolar protein subunits of 370 and 25 kDa covalently linked by disulfide bonds. Fibroin filament is made of both crystalline and amorphous domains. Sericin is a second type of silk protein which contains 18 amino acids including essential amino acids and is characterized by the presence of 32% of serine. The total amount of hydroxy amino acids in sericin is 45.8%. There are 42.3% of the polar amino acids and 12.2% of the non-polar amino acid residues. Sericin contributes about 20 to 30% of the total cocoon weight. Their main role is to envelop the fibroin. In presence of sericin, the fibroins are hard and tough and become soft and lustrous, and after its removal, sericin occurs mainly in an amorphous random coil and to a lesser extent in a beta sheet organized structure. The randomly coiled structure easily changes to beta sheet structure as a consequence of repeated moisture absorbing and mechanical stretching. The gene encoding the silk protein P25 is expressed in the posterior silk gland of the Bombyx mori with strict territorial and developmental specificities. The cis-acting regulatory elements previously located within the 441 base pair 5' proximal sequence of the gene were examined for protein binding capacities. Two factors were identified, BMFA and SGFP, that led to the prominent band shifts and the target sites for which are included in a region homologous to the fibroin gene enhancer sequence. Analysis of the tissue-specific incidence of both factors showed that BMFA is a ubiquitous, whereas SGFP is restricted to the silk gland cells. However, SGFP was found in both posterior and middle silk gland cells and therefore likely directs organ specific but not territory specific expression. Developmental studies throughout the fourth larval moult at which the P25 gene status changes from de-repressed to repressed reveal that BMFA is reversibly modified at the transition from the intermoult to moult. Indeed, the pre-existing BMFA is replaced by a structurally related factor BMFA. During the following head capsule apolysis, the exact temporal coincidence of this conversion with the onset of the gene repression suggests that BMFA is involved in the transcription inactivation and likely results from a transduction process initiated by the hormonal changes at molting. Let's see how P25 gene expression is made. The expression of the genes encoding the silk proteins in the silk gland of Bombyx mori constitutes an attractive model for understanding the molecular mechanisms governing space and time programmed transcription and quantitative tuning of mRNA production. One set of these silk gland specific genes with remarkable characteristics of the expression is the two genes coding for the silk protein P25 and fibroin, two abundant silk proteins of the cocoon. The P25 and fibroin gene expression is characterized by a restricted territorial specificity since it occurs only in the 500 cells of the posterior silk gland PSG and the cells in the median silk gland MSG express another set of genes encoding the so-called sericin proteins. A sharp developmental control of mRNA production is also exerted, making both genes highly transcribed during the intermol periods and repressed during the molting stages. The quantitative adjustments of P25 and fibroin production is essentially, if not exclusively, controlled at the transcription level. This includes a precise coordination of the rate of mRNA synthesis, which leads to the accumulation of the equimolar amounts of P25 and fibroin mRNA in the cytoplasm. The intricate mechanism that regulates accessibility of the genes to RNA polymerase II according to various cellular or developmental states are not understood yet, but they involve the binding of specific sets of regulatory factors to accurate cis-acting sequences, which may in turn trigger higher order complexes through protein-protein interactions. The DNA domain carrying the cis-acting elements of the P25 gene has been studied in vivo by gene transfer into Drosophila chromosomes. Secondly, the Bombyx gene promoter is activated in transgenic flies only in specific cells of the salivary gland, the organ homologous to the silk gland in which it reproduces its territorial and developmental specificities as well as its high transcriptional activity. All of these properties are specified by elements included within the 441 base pairs 5' proximal region. Interestingly, this domain includes a 4100 base pair domain shared by the P25 and the fibroin genes, which has been determined in vitro to be a strong potentiator of fibroin gene transcription and which has properties of an enhancer element. The description of the two distinct protein factors binding to DNA flanking the P25 gene is explained here. Their properties strongly suggest that they are involved respectively in the control of specific P25 gene expression in the silk gland and in the repression of its transcription at molting. The implication of these results in understanding the mode of regulation of the silk protein encoding genes is discussed in the figures A and B. In the following figure, A location of the different probes used for band shift assays in the P25 gene promoter. The open box delineates the region sharing the sequence identities with the fibroin gene enhancer element, and the arrow marks of the transcription start sites is being indicated. In figure B, footprinting analysis of BMFA and SGFB. Sequences protected from DNase 1 digestion are underlined by thick bars, and hypersensitive cleavage sites are symbolized by arrowheads. Thin arrows delimit diasymmetries in the DNA sequence between the two target sites. Major DNA protein interactions in the P25 gene occur in the proximal region of the promoter. With the aim of identifying the sites of the DNA protein interactions in the precise domain of the P25 gene that is sufficient to regulate its expression. Next, we will see the structural organization of the P25 gene. P25 and fibroin genes share clear-cut stage and cell specificities, high transcription rates, and also strict adjustment of transcriptional events since their mRNA are maintained in a 1:1 molar ratio during the period of silk production. The linkage of P25 to fibroin by disulfide bonds and the stoichiometry of their mRNA argue for an equimolar relationship between the proteins. The organization and structure of the P25 transcription unit in terms of size, plurality of the introns, nucleotide sequence of the 5' region do not correspond to those of the fibroin gene. This, together with the localization of the corresponding loci on the two different chromosomes, emphasize the absence of any obvious evolutionary relationship between the gene encoding the two subunits of the major silk protein. Contrasting with this observation, it is all the more remarkable that the flanking regions of the P25 and the fibrinogen gene share a high degree of sequence homology. That these may be significant with regard to the functioning of the P25 gene is strongly suggested by the fact that they correspond to the crucial regulatory regions that have been identified in the DNA upstream of the fibroin gene. From analysis of the sequences required for in vitro transcription, Suda and Suzuki have assigned a key role to two regions flanking the fibroin gene. One matches the quasi-universal promoter of the RNA polymerase II transcript gene, the sequence TATA25 that we find in the expected position in front of the P25 gene. It is noticeable that not only the TATA sequence but also several motifs surrounding this promoter are shared by both the genes. The other identifiable sequence, mainly between 239 and 116, function as a control element with strong modulator activity. Indeed, it corresponds to the region that was found highly homologous with the DNA upstream of P25 gene. Yet, the region of greatest homology from 230 to 142 in the fibroin DNA is shifted by about 100 nucleotide downstream in the P25 flanking sequence and is found between 138 to 49. Given the observations of Suda and Suzuki, that the effect of the distal element of the fibroin gene is not abolished either by the deletion of the DNA between the 65 and 44 or by an inversion of the region from 234 to 66. The shift of the equivalent sequence in P25 DNA may be of no consequence for its presumed role in the transcription modulation. Indeed, this would be compatible with the properties of cis-acting viral enhancers and certain eukaryotic regulatory elements, which can act at various distances from the capsid. A common feature of the P25 to 137 to 164 region and of the fibroin 231 to 155 sequence is that they contain dyad symmetry. Their potential stem and loop structure would be centered at nucleotides 99 to 194 respectively. This stresses the possibility that the conformational characteristics of the DNA may be decisive for the functional sequence to be reorganized by regulatory proteins so as to influence the efficiency of the basic promoter, the Tata box region. The two genes under study would thus be comparable to the Drosophila genes encoding the heat shock proteins, which share a conserved upstream promoter element that contains an inverted repeat. Dyad symmetry has also been found in the 5' flanking region of the Drosophila gene encoding the glue protein SGS4 and which is thought to be involved in the control of transcription efficiency. In a number of cases, the concerted induction of functionally related genes correlates with the presence of common short repeats in the 5' flanking regions of these genes. Direct evidence have been obtained from transformation experiments on the role of such sequences in the coordinate induction of the yeast, His and the Drosophila heat shock P70 genes. Consequently, sequences shared by the P25 and fibroin genes may be implicated in the concerted induction of their expression, which is probably hormonally mediated. Functional, in vitro and in vivo tests are currently being developed in our laboratory to assay the properties of the above sequence on the transcription modulation of the P25 gene. In addition, the question of whether some of these homologous sequences are unique to the P25 and the fibroin genes is being examined by sequence analysis of other genes expressed in the silk gland cells and in other tissues, such as the genes encoding sericin or cytoplasmic and muscular actins. Let us see the role of miRNAs in P25 gene regulation at post-transcriptional level. MicroRNAs, also called as miRNAs, are a class of non-protein coding small RNAs that regulate a gene expression at the post-transcriptional level. Using a silicon screening, it has been found that 3' untranslated regions of the P25 gene mRNA are perfectly complementary to the nucleotide 2 to 8 at the 5' end of the miRNA2B, MIR2B. The expression of the MIR2B and the P25 gene in the posterior silk gland of the 5th instar larval silkworm was investigated using real-time PCR detection method. The result indicated that expression of the P25 gene was very high in the posterior silk gland during the 5th larval instar, whereas the level of MIR2B sharply decreased until reaching the lowest one of the 8th day. The expression patterns of MIR2B and P25 gene indicate that MIR2B might act as a fine-tuning regulator of the expression of the P25 gene at the post-transcriptional level. Structural characterization of the individual silk components is a necessary prerequisite for elucidating their roles in the production, physical properties, chemical resistance and biological functions of the silk. The existence of the distinct class of small silk component, the seroins, that differ from the bulk of the large size silk protein structurally, and that their production occurs both in PSG where fibroins are produced and in MSG where fibroin coating by sericin is provided. This dual origin is emphasized by the term seroin. The secreted seroin. After the signal peptide removal, the seroin peptide of the Galleria differs from all known silk proteins by its high proline content, 34 residues or 20.26% by weight, and lack the cysteine. More than a half of the proline residues are contained in 2 copies of PPLPQ, PPPL repeat, and in 3 copies of PPI motif. Acidic residues prevail over the basic ones, and the calculated pA of the seroin therefore equals 4.93. The N-terminal amino acid sequence of the silk proteins reveals that the seroin peptide is present in silk as 22.5 and 23 CADA glycoproteins. Their reactions with concavalin A indicates presence of mannose-rich carbohydrates. The nanoglycosylated form of seroin, which would migrate as a 16 CADA protein, is not secreted. This situation is reminiscent of the P25 gene product that also occur in the silk as 2 mannose-rich glycoproteins whose apparent size is by 6 to 7 kilodaltons, larger than the molecular mass of the detected peptide. The relatively high content of polar amino acids and the glycosylations should make seroins water soluble. However, washing silk with hot water did not reduce the content of 22.5 and 23 CADA proteins. This observation suggests that seroins are liberated from the silk only when most of the silk components are dissolved. The function of seroin is elusive since the high proline contents is reminiscent of the proline-rich antimicrobial peptides. We suspect that seroin may be responsible for the silk resistance to molds and bacteria. Let's see the seroin gene and its expression. Research studies indicate that there is one copy of seroin gene present in the genome, similarly to other Lepidopteran silk genes. As in other silk genes, cerein expression exhibits a high degree of spatial and temporal specificity. However, the simultaneous expression of the cerein in PSG and MSG is unique. Downregulation of silk genes by ecdysosteroids was previously illustrated in experiments with isolated abdomens of post-feeding Galeria larvae. The removal of the head and thorax deprived the insects of their source of ecdysosteroids and most other hormones, but abdomen survived and the silk glands remained functional for many weeks. The contents of the sericin mRNAs and P25 mRNA3 declined gradually in such preparations, but applications of mold-inducing 20E dose elicited their rapid disappearance. These experiments could not reveal, however, if 20E acted directly on the silk gland or if the effect was mediated by some other tissues. For example, the fat body that provides silk gland with nutrients. In the present study, it shows that the content of cerein mRNA declines when explanted silk glands are exposed to 20E in the absence of any other tissue. As expected, the decline is much lower in the presence of protein synthesis inhibitors, indicating that the cerein gene is not the primary target of the hormone. The titer of the ecdysosteroid rises and the activity of the silk glands is diminished during each ecdysis. Earlier, it was considerable amounts of sericin mRNAs persist in the silk gland during the larval ecdysis but becomes undetectable during the pupal ecdysis. When the silk glands initiate apoptosis, administration of a JH analog to larvae before pupal ecdysis prevents apoptosis and the sericin mRNA remains present. It now shows that JH exerts such a protective effect on the cerein mRNA in explanted silk gland in the absence of any other organ. The results of in vitro tests demonstrate that both ecdysosteroids and JH act directly on the silk glands. An enhanced ecdysosteroid concentration suppresses the expression of the cerein and other silk genes, and in the absence of JH, it elicits silk gland degeneration. The latter effects is prevented and silk gene expression is restored when exogenous JH or a JH analog is added. Thereby, we conclude the chapter on the regulation of silk encoding genes. Thank you for being with us and hope you enjoyed the session.